Our next speaker is Dr. Christopher Bunker. Chris is Vice President of Business Development at Advanced Cell Diagnostics. Chris earned his doctorate from Harvard University and has an MBA from Boston University. He completed a postdoctoral fellowship with the Leukemia Society of America at the University of California. Prior to his current position at Advanced Cell Diagnostics, where he leads its assay development service business unit, Chris has worked at Genomic Therapeutics Corporation and Cell Signaling Technology. Chris's talk today is entitled Expanding CDX Strategy Options with RNAscope, a clinically validated, quantitative, in situ RNA biomarker platform. Chris, welcome.
Good afternoon, thank you. Advanced Cell Diagnostics is a company which has been in business for about 7 years now. We were acquired last year by Biotechne. We're a research use only products company. So we're definitely kind of on the fringe with sort of putting ourselves in the context of what Oscar talked about yesterday. We do not have any Super Bowl rings, not by far. We are trying to work on his analogy. I was thinking the one-handed catch. Odell Beckham, he's most well known for the one-handed catch, and that's become pretty standard now in NFL. But for a long time, no one thought that was possible. So the analogy is we have a new technology which is based on in situ hybridization, RNA in situ hybridization, which has been around for decades, but no one does it because it never really worked. It was not reliable. It was all homemade stuff. This is a new, vastly improved technology, which is gaining a tremendous amount of traction. Again, we've been in business for 7 years now. We've grown 50% year over year, every single year. There are over 1,000 publications now, and there are, in fact, companion diagnostics in development, being used or will be used next year by Bayer Pharmaceuticals and Merrimack Pharmaceuticals for registrational clinical trials. So this technology has been chosen in those cases because it outperformed IHC, qPCR, and NanoString for correctly and robustly, rigorously identifying patients that respond to their therapeutics. So that's, in a nutshell, our story. My presentation will mostly be sort of to introduce technology, which hopefully will sit in your minds as a possible option. Right now, we're trying to primarily position it as an option, not the be-all and end-all, but it has a lot of advantages in comparison to other technologies, IHC methodologies, or other molecular extraction-based technologies. So again, this information I've already told you, a lot of publications. We have so our technology is analogous to NanoString in terms of its oligonucleotide probes. It doesn't have the advantage of being able to do such high levels of multiplexing, but as you'll see throughout the presentation, it has a major advantage of putting that information, molecular information, in the context of intact tissue. So you can look at expression within a specific cell type and quantify that expression. So we offer both products, which are used throughout biopharma, academia. We also offer assay services. Our services in relation to biomarker development are definitely early stage protocol development. We are not a CLIA lab. We do not do patient testing, except for retrospective testing to, again, establish assay performance, demonstrate its performance against other methodologies. Then those assays would be then transferred to a CLIA lab for use prospectively. But again, the advantage of this methodology in comparison to other tissue-based assays is it's single molecule sensitive. It has the specificity of a molecular test like a NanoString assay, again, oligonucleotide based, relying on multiple oligonucleotides, which improves the specificity of the assay, and the information is provided with single cell resolution. So we can quantify expression cell by cell by cell, whether it's in immune cells, peritumor adjacent stromal cells, tumor cells, whichever, that can be quantified cell by cell, and it can be multiplexed to a limited degree. And so with that, again, we now have tools for providing early stage target validation, where there aren't any other reagents available, and again, biomarker development. The speed with which we can develop assays is another major advantage. Just a couple of weeks and we've got something ready to use, and we can be doing the validation studies very quickly from that point on. So with respect to our potential use of the assay as a diagnostic, it's in thanks to our partnership with Leica Biosystems, which is the commercial channel for RNAscope-ish based diagnostics. So we are the manufacturer of GMP-compliant reagents, materials being used by Leica for developing kitted RNA in situ hybridization-based tests. So they're offering now both standalone ASRs for just some low-hanging fruit, standalone diagnostics, viral CMV, EBV, albumin in liver, and they're also the ones developing companion diagnostics again for Bayer Pharmaceuticals and Merrimack Pharmaceuticals. And so just to kind of give you some alternatives to what you've been hearing, definitely won't see any statistics like were shown on the previous slide, but rather just some images to give you an idea of what this data looks like. And again, every time you see signal with the RNAscope methodology, again, we're detecting individual molecules of RNA. So dot by dot is measuring transcript by transcript in a cell-specific manner. In this case, showing markers. Heregulin, NRG1 is is one of the diagnostics being developed for Merrimack Pharmaceuticals. This is the ligand growth factor of ERB3. This is the biomarker that identifies patients that respond to Merrimack's anti-ERB3 therapeutic in non-small cell, ovarian, and breast cancer. It's not the target itself, it's the ligand which drives signaling through that pathway, which is the biomarker. PD-L1, of course, this is to illustrate, and I'll show you another example of the level of expression that can be detected with this methodology is far below anything that's achievable by IHC, simply because, again, we're detecting individual molecules, whereas it requires tens of thousands of molecules of protein to be detectable by an IHC assay. We can do this now in duplex. Here we're showing just examples of combining PD-L1, CTLA4, or IDO1 and interferon gamma cytokine expression in this greenish-blue signal and red signal. The advantage of this methodology, again, oligonucleotides, we can mix and match them in any combination. Any combination of probes that make sense to look at in combination can be done simply by pooling different probes together. Again, not on the order that you can achieve with nanostring technology by any means, but the fact that we can put this information in the context of intact tissue, tell you where are different immune cell populations, where is cytokine expression happening, where is which subpart compartments of the tumor are actually expressing the target. That information can be captured all within an intact tissue section. So I won't go into too many gory details, but this is all based on, again, oligonucleotide probes, all of which hybridize exactly the same way to their target transcripts. Again, with the nanostring presentation yesterday, that's based on using two oligonucleotides working together to both as a means of capture and a means of detection. We use multiple oligonucleotides, but it's two together that result in signal amplification. So we're not enzymatically amplifying signal. We're amplifying signal through sequential hybridization. Everywhere that a pair of oligos hybridize to a target transcript, those then will hybridize to other oligonucleotides, again, all very much localized, so that you get a localized sort of compilation of molecules of label, in this case either HRP or alkaline phosphatase or fluorophores, but all that signal ends up being in one physical location. And so we use a lot of oligonucleotides, knowing that this target transcript is highly fragmented, not generally accessible. We use a lot of oligonucleotides to ensure that we have a very high frequency, a very high probability of detecting every molecule in that tissue section. Not absolute, just like any molecular assay, it's not absolute, but as sensitive, if not more, than nanostring or qPCR. And we've shown that, and I'll give you examples of that in a moment. So again, it starts with FFPE tissues, conventional tissues, archival tissues. We've used this on samples 10 years old, 20 years old. We don't necessarily recommend using 20-year-old tissue, but it works on tissue if it's been properly fixed and stored. Any FFPE tissue will be suitable for analysis with this methodology. And so what happens again when oligonucleotides hybridize to the target transcript, we're then sort of piling on a tremendous amount of label onto that single transcript, which gives us a means of detecting that in a fixed position. And these assays run fully automated on Leica as well as Ventana autostainers. But again, to emphasize this versus when you think about an IHC assay where you have a primary antibody binding to a single epitope, presumably it's monoclonal. Then you come in with a secondary antibody, and that's maybe you stripped out and biotin. You get a few molecules of label per protein, which means you need, again, tens of thousands, if not hundreds of thousands of molecules of protein to generate a detectable signal. Whereas here, this generates signal which corresponds to a single transcript of RNA, which gives us an assay which is going to be orders of magnitude more sensitive than IHC. And so again, this can be quantified counting expression cell by cell within intact tissue. So in terms of the development of potential diagnostics and tests that are used to identify and select patients in clinical trials, are part of the, we develop assays. We develop assays on the research instrument of Leica, their Bond RX system. This is essentially identical to their diagnostic FDA-approved instrument, the Bond 3. This is what diagnostics will be run on. We can develop the assay on the Bond RX and then very easily transfer that to the Bond 3 for diagnostic use. So this is obviously a somewhat biased perspective on the utility of RNAscope versus other methodologies, but I think it's largely accurate. First and foremost, morphology. If morphology doesn't matter, then forget it. Grind it up, and if a molecular signature is going to tell you what you need to know by all means, that's the way to go. Highly sensitive, easy to do. If morphology matters, if knowing which cells are expressing what, then we have significant advantages over assays that don't provide any morphological context. Sensitivity and specificity, I think almost invariably we will be more sensitive and more specific than any IHC assay. Just because of the molecular detection and the fact that we're dealing with nucleic acids, which are predictable. We always know how a nucleic acid will hybridize. You have no idea how your antibody binds to your target antigen by in an IHC assay. And in fact, the tissue requirements for our assay are quite minimal. Oftentimes, for molecular extraction-based methodologies, you need a substantial amount of tissue. For ours, we need a single tissue section of 5 microns, just like IHC. Again, the advantages, light microscopy or fluorescent microscopy to look at expression in intact tissue, single molecule sensitive. It can be pathologist scored or using software for quantification, and it's a fully automated instrumentation platform. So I won't spend too much time on this, but this is a publication, one of the first publications with RNAscope back in 2012, just showing if we count green dots, which in this case correspond to HER2, or measure those in solution, this being a standard curve of in vitro transcribed message, we get about the same number, 17 transcripts per cell on average in an extract versus 14 dots per cell if we simply count them by eye, which tells us that when we see dots, that corresponds to a transcript. Again, not an absolute measure, but every time we see a dot, that corresponds to a measurement of a transcript. And this is some data from breast cancer tissue measuring HER2 again, or in this case, heregulin from homogeneous populations of cells expressing heregulin at different levels, showing very good concordance between qPCR and RNAscope. So we have good concordance by that methodology. And then this is actually some really recent data, which, again, nanostring, tremendous technology, tremendous potential in terms of the sensitivity and multiplexing capability. But here's just an illustration of this is an average dot per cell as measured with quantitative software versus a nanostring score, basically down to the very bottom limits of detection with nanostring. But if you look at the image, you can clearly see those signals. A signal which, on average, out of all the cells in the sample, very few transcripts per cell, but among the few cells that express, high-level expression. So that's the advantage of a molecular assay put in the context of intact tissue. And there again, same thing. In this case, now detecting secreted factors, IL-1B or TNF, which not going to be detectable by IHC, very low level as measured by a molecular extraction-based assay, but put that in the context of intact tissue, you can see which cells are expressing, how many cells are expressing, where they are, and what their actual expression level is. And then this is, again, really to emphasize the fact that it's like any other molecular test or assay, designing probes for detecting anything that looks interesting, any biomarker, any new target. Developing those assays will take a matter of weeks. They're ready to use, and you can be generating tissue-based analysis data very quickly to dovetail with discoveries perhaps made by transcriptomic approaches. And so in terms of, again, as a sort of benchmarking against IHC, which is still our primary sort of competition or the standard sort of gold standard biomarker assay in immuno-oncology is still at this point. Our assay is going to, again, be substantially more sensitive. This is just one illustration. Collagen 11A1, a biomarker of ovarian disease progression, showing detection by RNA versus detection by protein. They show concordance in terms of both are upregulated. The signal coming from an RNA assay is substantially greater because, again, we can detect many fewer molecules as compared to the amount of protein you need to detect with an IHC assay. And then likewise, in comparison to diagnostic tests for PD-L1, we don't necessarily expect RNAscope to be used for PD-L1 testing. That horse has probably pretty much left the gate, but we can show that it does provide an assay with much wider dynamic range because of the higher levels of sensitivity. So perhaps if this was to be revisited, or someone comes along with another PD-L1 drug and we can be involved in that clinical trial, we are likely to be able to show them a more precise cutoff of where patients start to show responsiveness as compared to less sensitive assays like IHC. And so I mentioned it's a quantitative assay. We use software from Halo, Indica Labs, Halo software for doing cell-by-cell quantification of 1, 2, or 3 markers simultaneously. And that's, again, information we can capture that information from an entire TMA core or entire tissue section to understand what percentage of cells are expressing, where those cells are in relation to each other, much like you can do with IHC, but again, with a more sensitive assay, more specific assay, and therefore a more quantitative assay. In terms of a diagnostic platform, there are many technologies. We are now one of those. We're being utilized by a number of companies. I've mentioned Bayer and Merrimack are the two we can speak about. We have about a half dozen other programs that we're working on currently. In most of those cases, yeah, we were the last resort. We do still represent a significant risk as compared to a Ventana IHC assay or even a nanostring assay. But when it comes down to it, pharma companies are running out of options for some of the immuno-oncology targets. They know us through use in preclinical research, and we can very quickly show the performance of our assay for measuring IDO1, TIM3, LAG3, PD1, PDL2, any of those markers, we can show them the performance of our assay against whatever other methods they're considering. So very quickly, we can provide a means of comparing and contrasting how RNAscope might provide an alternative which may or may not be a better solution. So just to speak about the two assays which are now in development as companion diagnostics, provided the clinical trials continue to go well. For Merrimack Pharmaceuticals, they were one of the early adopters of our technology. That's primarily because their therapeutic strategy is to develop antibody therapeutics that block receptor tyrosine kinase signaling pathways in cancers where those pathways are mediating chemoresistance. So they're looking to block signaling pathways that will then resensitize those patients to chemotherapy. So in all cases, it's going to be a combination of standard chemotherapy plus in this case, this is MN121, their anti-ERB3 therapeutic. And it turned out it was the ligand, or heregulin, which looks like this in a tissue biopsy, that predicted responsiveness. In their phase 1 clinical trial, they used this retrospectively and identified patients, which here, the subpopulation of patients in breast cancer, ovarian cancer, and lung cancer, which had high levels of heregulin, those are the ones that showed responsiveness to an anti-ERB3 treatment in combination with chemotherapy. They then used it in a phase 2 trial as a selection criteria for enrollment, and that clinical trial was successful, and now they're planning a phase 3 trial where that, again, will be the basis for registration. The clinical trial will be run at LabCorp, Covance LabCorp with a kitted test developed by Leica Biosystems. And then the other one, this sort of relates to the previous talk, but speaks to the fact that actually for FGFRs, it was shown a couple of years ago that gene copy number amplification, genomic changes in FGFRs does not predict responsiveness to small molecule tyrosine kinase inhibitors targeting that family. And in fact, expression, whether it's protein expression or RNA expression, is much more predictive of sensitivity to tyrosine kinase inhibitors targeting FGFRs. And that's the basis for Bayer Pharmaceutical using that in their phase 1 clinical trial. And in this case, they compared, again, we weren't their first choice. I'm sure they tried all the available antibodies. and they also compared it to NanoString. So we were put head-to-head against NanoString. They chose RNAscope as their companion diagnostic because it showed a higher level of specificity. Both were very highly sensitive. Our assay proved to be more specific, and that's the basis for their decision to have Leica Biosystems develop that companion diagnostic. Again, we're still pretty early stage. We're being used in more and more clinical trials, more as an exploratory marker. But again, wherever we've been put head to head with other methodologies, we've shown to we've been able to outperform those in terms of a metric or a methodology that will selectively, accurately select patients for clinical trial enrollment. So I'll leave it at that. Thank you.